Okay, our next speaker here is Julie Larson. Julie's a second-year master's student at Oregon State University with a strong interest in plant ecology, invasion, and restoration. Her graduate research has been in collaboration with some folks at USDA, ARS, and Burns and Boise. She is originally from the Midwest, but she has actually grown fond of life out here, and will be looking for continuing to work in the area of either management research or extension following her rapidly approaching defense date in August. So please join me in welcoming Julie.
Good morning. All right, so as Jeremy mentioned, I'm a graduate student at Oregon State, and I'm also working with the USDA. So I'm going to be talking to you today about a little piece of my graduate research. And this is actually the first time that I'm presenting any of these preliminary results to the public, so this is exciting. I'm excited to share some of these things with you and get you thinking about these ideas. The other speakers this morning have really set me up well talking about this seed and seedling stage of recruitment, giving you some of the background in terms of how the environment might influence success during recruitment at different stages of emergence and germination. And so I'm hoping to tie in my little piece here. Hopefully your brains aren't too exhausted with science, but to bring traits into the picture, to understand how traits might also influence success during recruitment. So with this project, we had 47 different varieties of grasses that are found in Great Basin ecosystems, and we measured seed and seedling traits on these 47 varieties to try and understand the different recruitment strategies that exist in these systems and identify groups of species which are similar. And in doing that, we were really also interested in this origin aspect to understand whether there were some real differences in recruitment strategies between native and introduced species, and then also annuals and perennials. And we also were interested in the effects of water and whether water had an influence on these trait trends that we were seeing. So I'm going to give you a little bit of a background in recruitment so I can also explain how traits play an important role in the process of recruitment. And then I'm hoping to spend most of the time talking about our specific research questions and some of these preliminary results. Mind you, we're just getting started with the analysis, but we're seeing some interesting things. So just to revisit this process of recruitment, you saw a similar slide in Jeremy's presentation earlier. Recruitment is basically, in general, the process of an individual reaching this mature status, transitioning out of the juvenile period. And as Jeremy pointed out, we recognize that there are actually several different life stage transitions that these individuals have to pass through in order to successfully recruit. Jeremy talked about what these are, but I also want to talk a little bit about the challenges that exist at each of these stages. You start thinking about the challenges that each individual faces during these life stage transitions, you can start to think about what traits might be important in determining whether or not they're successful. So you get a seed in the ground, it has to be able to successfully germinate. And as Nathan was pointing out, different seeds may have different moisture and temperature requirements. That moisture and temperature is really important in determining whether or not a seed successfully germinates. So in order to germinate, the seed has to have those conditions. And then once a seed germinates and it's sitting there in the soil, it has to be able to survive until it's able to emerge. And Jeremy talked about that as being an important bottleneck that he found in Eastern Oregon, where these seeds were germinating, but they were dying before they were able to emerge and start photosynthesizing. And we're not exactly sure why that is. Trait differences between species could be a good direction to go to start to understand that. Even then, when a seedling emerges, that coleoptile comes up out of the ground, it's incredibly vulnerable. It's a small root system, temperatures and conditions could be fluctuating, and it has to be able to survive long enough to develop true leaves and begin photosynthesizing independently of its seed reserves. And eventually, even when it starts photosynthesizing, there is a huge literature base that talks about how vulnerable seedlings are in other systems around the world. We know that adult plants have larger root systems, they have more leaf matter, they're just better equipped to survive things like drought and herbivory than seedlings are. And so seedlings still have this challenge to overcome in order to successfully recruit. So this is, hopefully you understand why recruitment is such an important process. We know that not only is this a vulnerable life stage in general, but that mortality can actually occur in between all of these life stages for different reasons, potentially. And if we know that recruitment is a limiting factor for these plant populations, we might start to think that, for instance, species that are failing during recruitment are potentially, or recruitment failure is potentially a reason why our restoration is failing. By the same token, maybe species that are more successful in recruitment are more likely to be able to be restored or will be more successful in the long run. So if we can understand out of our whole suite of species that we've got out there what the different recruitment strategies are and how species differ, we might be able to understand what is driving differences in recruitment success. So this is a pretty cool piece of the puzzle. It's very logical and mechanistic in my mind. So I want to talk a little bit about the importance of functional traits to give you a background of why this all should make sense in theory. Functional traits, there are pretty much any characteristic of a seed or seedling could be a trait. The definition of functional traits is a little bit more specific. They are, in theory, supposed to have some sort of direct influence on the growth or survival of an individual. So functional traits, in other words, functional traits define an individual's ability to succeed in its environment. And this is especially true of plants because plants are stuck in one place, right? So they can't get up and move. They're sort of stuck with either the water availability that they have, the nutrients availability, temperature. And the traits that a species has in order to collect resources or conserve resources or to germinate in the first place, the traits that define those abilities are going to define its fitness and its ability to actually survive and succeed. So if we accept this notion that a plant's ability to survive is directly linked to its traits, then it follows that traits could provide a common currency for understanding relative fitness differences across species. All of the grasses in my study are grasses. They all have similar traits, but they differ in those trait values. And we're hoping to show that eventually show that there might be a link between those differences in trait values and differences in recruitment success. An important note, Tom touched this morning on the fact that we've been studying cheatgrass for decades. And this isn't a new notion at all. People have been studying cheatgrass and its traits for a really long time. We know a lot about cheatgrass. We know that it grows really quickly, that it produces a lot of seeds. And a lot of studies have been done comparing cheatgrass to a few of the traits of a few of the dominant native perennial species. And it's really tempting to say, okay, well, we see this difference between cheatgrass and this one native perennial bunchgrass, so maybe these traits are driving cheatgrass's success. But until we expand our sort of like data set across species and really get the full spectrum of traits that are out there, we can't really pinpoint recruitment success to a particular trait. We can say cheatgrass was more successful and it has these traits, so maybe that's the reason why. But what we're trying to do with this research is to really use enough species and collect enough trait data that we can talk about traits and how traits influence success. Because if we can turn it, instead of talking about species, if we can bring it to the trait level, then we could use it as a predictive framework to predict how well any species is going to do in our systems and really understand not only, in order to understand why maybe they're being more successful than others. So. And one last point, like I mentioned, there are a lot of traits out there. The key thing is that many plant traits are known to be highly correlated, and that suggests that we might be able to detect overall strategies. So not just a seed trait of a species and a seedling trait, but maybe those are actually related, and we can define whole recruitment strategies, if you will, and that will help us sort of group species which are more similar to each other. So that begs the question, what traits do we think might actually be important during recruitment? And there are more traits out there. These are ones that we measured that I'm just going to briefly introduce you to, but there are more out there. So this pointer thing would be helpful. So during recruitment, we actually have these very different life stages where individuals exist mostly as seeds and then mostly as seedlings. So I've sort of grouped these out into pre-emergent traits and seedling traits. You'll see later that actually, some of the trait values are related between this group and this group, which is interesting. But these first 4 traits right here are all germination requirement traits. They all come from the hydrothermal time modeling that exists out there that Nathan talked about a little bit earlier. Base temperature for germination and base water potential for germination basically describe minimum moisture and temperature requirements that a seed needs to germinate. And thermal time and hydrothermal time reflect how long it takes a seed to germinate given moisture and temperature conditions. As conditions for germination become more favorable, as temperatures are higher and moisture content is higher, the difference between seeds that have high hydrothermal times and low hydrothermal times will tend to germinate will take the same amount of time to germinate if conditions are very favorable. But during that in-between process where maybe temperatures are a little bit cold and things are a little bit dry, these differences in thermal time and hydrothermal time could mean huge differences in when a species actually germinates and how long it takes. So just to touch on some of the others, I wanted to explain that since they're not very commonly reported or used in the functional trait literature yet. But we also looked at seed mass, coleoptile tissue density, which we're hoping is sort of a proxy for how tough this tissue is when a seedling first emerges. And then over in the seedling traits, these are all traits that have been measured forever in the adult plant trait literature. And we know that there are some pretty good relationships between things like specific leaf area and relative growth rate. Species which have thinner leaves tend to grow a lot faster. They photosynthesize more quickly. And so we looked at a few root traits here and some leaf traits, and root mass ratio, which basically describes how much of that mass is being allocated below ground from the whole plant. And yeah, we'll talk a little bit more about what that might mean in terms of where the trade-offs are and implications for fitness as I go through the results. So just to recap the research questions that we're going to be addressing today. This first one, sort of the main questions. Can we identify distinct recruitment strategies based on these pre-emergent and seedling traits? We're asking what kind of functional groups emerge in terms of species which are more similar to each other, and which of the traits are really driving that group differentiation. Then we want to ask whether these trait-based functional groups align with our previous notions of how we're categorizing species in the system based on their native and introduced status and their annual perennial status. And that's a really interesting question because we have in the past used these other categories in management. And finally, I'm not going to probably present any of the specific results from how water addition influenced these trait strategies, but I will talk about, as I'm going through the results, how adding water changed the things that we saw, so I'll make sure to comment on that. It's another important question. Okay, so this is the fun part where I actually get to talk about the project and the results. So this is my study site, and you may notice that it looks a little bit familiar from Jeremy's presentation. That's the same, the butte, we like to call it in the background. I don't know, I guess it's kind of hard to see me. That's actually me right there. I think I was laying there for the purpose of scale, but that's actually how I felt many days after spending an entire day in the field. So this was out in Eastern Oregon. And we, like I said, we used 47 semi-arid grasses. There aren't actually that many species that we were able to identify, but we used multiple varieties of certain species. And I know that probably a lot of you are going to wonder what species I was using, and you'll see a list later. For right now, I want to keep the focus on the traits, but I had 4 introduced annual grasses, including cheatgrass and Japanese brome, and then the rest of them were split between native and introduced perennial grasses, everything from squirreltail to crested to blue bunch to the needle grasses, fescues, and a few other less common species, some perennial bromes too. And we collected our seedling traits from individuals that were measured in the field. They were grown in these bags. So we collected them. We actually collected these seedlings to measure traits at 2 different time periods. The first one was right around this period of seedling establishment, and then when we went out and collected these bags and brought them back to the field, or back to the lab, sorry, we left half of them in the field for an additional 3 weeks, just 1 seedling per bag. We came back in 3 weeks in order to collect relative growth rate and also see whether any of these leaf and root traits had changed over time. And like I said, there was a watering treatment applied to these seedlings. Some of these seedlings received additional water from March to June. And our hydrothermal time estimates, I have to really thank Stuart Hardegree for this one. He housed me in his lab for about a month and a half where I sat there every day and counted 2,800 of these, which I don't know if it's a little hard to see. Each one of these has like about 30 seeds in there, and we essentially just had them going at different temperatures and water potentials and counted germination every day. Used some very simple hydrothermal time modeling techniques to come up with those parameters I talked about earlier. An additional note, you'll see all these plots. So those seedling bags are kind of in the rows here. They were just grown out in this field. And in these plots, we also followed some different demographic things like germination percentage and emergence percentage and seedling survival. And I'm not going to talk about those results, or I'm not going to tie the traits to that today, but that's something that we're hoping to do, is eventually tie traits to those demographic transition rates that we measured. So I just want to recap that these were the traits that we measured, and I want to point out, so we had 2 harvest times here, so altogether we had 19 different traits in this models that we were doing. And the reason I want to point this out is it's really hard when you have 19 different dimensions of variation among species, 19 different ways that species can be different. You need a way to sort of condense that down into something that makes sense, right? It's hard to think about things being different in 19 different ways. So that's where this TCA analysis is really useful. So before the analysis, the variation among species is spread. You can think of it as being spread out across 19 different dimensions of trait variation. And what PCA does is it uses high correlations between traits to reduce all of that down to just a few dimensions, usually 3 or 4, of species that of traits that are highly related. And we refer to those as axes, so axes of trait variation. The first axis always explains the most variation among species. Usually the most correlated traits fall onto that axis. They're most highly correlated. And then axis 2 takes whatever remaining variation there is and uses traits to explain that remaining variation, and so on and so forth. So we ended up with 4 axes of trait variation, which I will go through now, actually. So you can think of all of that trait variation that I talked about as 100% of the difference among species. And so this percentage right here basically describes how much of that 100% of the variation among species is described by each of these, what I like to call trait spectrums, each one of these axes. So the first axis that came out ended up being a bit of like a growth axis. On one end of the spectrum, we had species with really high SLA and high relative growth rates. And on the other end, we had species with really high leaf dry matter content. So really thin-leaved, fast-growing, and really sort of thick, tough-leaved, slow-growing species over here. And that's really common in the literature for adult plants, and we're seeing it here among seedlings too, that trade-off exists. Interestingly, we also had seed mass and root mass coming in on this axis. So species that have tougher leaves are also the larger seeded species, and they also have higher root mass. An interesting thing is just at this T0 phase, right around seedling establishment. Not when we went back and measured root mass at T1 3 weeks later, these species that had this high T0 root mass did not necessarily have that 3 weeks later. So it's really the correlation with this initial root mass just right around seedling establishment. The second axis that came out that really differentiated species was size. And this is really pretty easy to understand. On one end, we had species that had really large seeds, really large roots, and on the other end, very small seeds and very small roots. And that's pretty straightforward. The third axis that came out was interesting. It ended up being similar to this up here. We had this sort of leaf economics spectrum. down here we had this root economic spectrum come out, where species that had species on this end of the spectrum allocated a lot of their total biomass to their roots. So they put a lot of their carbon into their roots. Whereas species on the other end had high specific root length, meaning maybe they didn't invest as much in their roots, but for what they did invest, they got more root length out of it. So more bang for their buck, if you will. And finally, this last axis that came out were the germination requirement traits. And this described the variation in species in terms of their hydrothermal time requirements. So on one end, we have species that basically take a long time to germinate. You remember Nathan talking about Great Basin wild rye, how that really took a long time to germinate compared to those other perennial species. I had a rough time with that one too, actually. Yeah, that one took quite a long time in the lab to get there. Whereas, cheatgrass on the other end of the spectrum is germinating very quickly in a given temperature and moisture scenario. But it turns out, so species on the other end of the spectrum, we have species with high base water potential. So basically, they need a lot of moisture to germinate, but once they reach that moisture threshold, they germinate really fast. So, and interestingly, we have high specific root length at the T1 stage come out on that axis too, which suggests maybe there's some correlations between how species are germinating and then what they're like as seedlings. So that's pretty cool. And altogether, these 4 dimensions explain about 67% or 2/3 of the trait variation among species. This is a really cool set of trait spectrums, I think, because we can use these now to really understand how species are differing. Each one of these axes is independent of the others, meaning that species might be on this end of the growth spectrum, but on this end of the size spectrum. And so by using these 4 different spectrums, we can really understand how species group out with respect to these 4 different dimensions, if you will. We saw these similar axes come out with the water data too, so that suggests that these are hopefully pretty robust axes of trait variation. So now the really cool thing is, like I was saying, you can use these to figure out which species are more similar to each other on these 4 different axes, which is what we did next. So this is what it looks like when you plot the first axis along the second axis. So this was our growth axis down here, if you remember, and our size axis along this y-axis here. So species which are more close together, like this cluster right here, tend to have both similar growth strategies and similar size. And actually these are small-sized grasses, little poas and fescues that came out together down here. And so you'll see, what I want to point out here, we have a key for the introduced annual, perennial, and then the native perennial grasses. And it's kind of hard to see, but I'll show you. The introduced annual grasses all came out like right over here, way on the side of the spectrum that has high SLA, high root growth, relative growth rate. And so you can kind of see that a lot of our introduced perennial species came out up here, and a bunch of our native perennial species sort of came out down here. So it does sort of appear that, at least in terms of growth and size, that there's some differences between our introduced annual and perennial and native perennial species. But we wanted to go a little bit further, and we used a cluster analysis that actually looked at not just these 2 axes of variation, but all 19 different traits, and grouped species that were more similar together based on all of those traits. And we ended up identifying 6 groups of 6 functional trait groups that I'll go through in a second here, of species that are relatively similar in terms of their traits. So there they are. It's a little bit hard to see the coloring, but I'll go through these one by one. So first, over here, we have granted, I just came up with these titles for these groups a couple of nights ago, just to try and help us remember how these species are actually differing, like what traits are setting them apart. So these aren't set in stone titles, but they're helpful. So these are what I like to call the fast developers. We have our 3 of our 4 introduced annual grasses over here, but also, interestingly, several introduced perennial grasses and native perennial grasses falling out in this group. They tend to have high, like I said, high specific leaf area, high relative growth rate, and also very low thermal and hydrothermal time requirements for germination. So they're germinating pretty quickly and also growing quickly as seedlings. Then in the middle here, this is supposed to be pink. I called this group the average bunch for bunch grasses, average bunch grass. So I know, it was late. It was really late. These fell out pretty much, as you can tell, smack dab in the middle. So if you think about it like relative to all of the trait strategies that exist out there during recruitment, these guys were right in the middle. And these actually tended to be some of the elemuses, and some of the blue bunches fell out in this group, mostly native perennial grasses, right in the middle there. And they had low to moderate relative growth rate and SLA, and moderate root and germination requirements, or traits and requirements. Then down here we had these small seedlings that were really set apart by their small seedling size. So fescues and poas, Sandberg's bluegrass. They tended to have very small roots, obviously, but interestingly, very, very high specific root lengths. So these roots were tiny, but they were using the little bit of mass they did have to get longer roots. They had very high base water potential and thermal times, so they had very large moisture requirements, and they also just took a really long time to germinate in general.